In this practice session, we will learn how to use several tools related to cancer genetic background and genetic susceptibility to develop cancer. BRCA1 and BRCA2 are well-known tumor suppressor genes related to DNA repair, which are frequently mutated in various cancer types. It turns out that mutations in these genes are often inherited, resulting in substantial increase in probability to develop breast, ovarian, and prostate cancer throughout lifetime. Let's get back now to the Census Gene website, as some useful information can be found there in this regard. We will search for the keyword BRCA. In the results table, we can see a set of genes which their name or description includes the phrase. By looking at the table, we can see that germline mutations were indeed reported in the BRCA genes, as well as in several other genes such as BAP1, BARD1, BRIP1, and PALB2. All these genes form the BRCA-associated genome surveillance complex, BASC. Germline mutations in BRCA1 and BRCA2 are more frequent in the population and are therefore account to more predisposition cases. Where can we find information regarding frequencies of specific variants in the populations? The most important available resource today is the GNOME-AD site, developed at the Broad Institute. GNOME-AD is a resource which includes sequencing data contributed by a large number of researchers worldwide, with the goal of aggregating exome and genome sequencing data, making summary data, in particular allele frequencies, at each position in the genome publicly available for the scientific community. The site integrates data based on distinct populations and currently integrates data for more than 140,000 individuals. The interface is very simple and intuitive. The site can be searched by gene name, variant coordinates, SNP IDs, and more. Let's search with BRCA1. We can see now the structure of the gene, and in the table below we can see all variants reported in the population within this gene. Let's be more specific now and search for information regarding one particular variant, deletion of AG in position 185 in BRCA gene. This variant is well documented to confer breast and ovarian cancer predisposition. The deletion of two nucleotides early in the transcript causes frameshift mutation and non-functional protein. The genomic location of this variant, according to version AG37 of the human genome, is appear now on the screen. Let's search for variant reported in GNOME-AD in a small window around this site. We can see a short list of 8 variants in that span. Most of them are missense mutations and are very rare in the population, as we can judge by the absolute number of observed alleles and by the allele frequency. But our variant, the frameshift variant, cannot be considered as a polymorphism with allele frequency of only 0.02% in the general population, but is relatively frequent considered to the other variations. GNOME-AD integrates sequence data based off individual sequencing projects of distinct populations. By clicking on the variant in the table, we can see the allele frequency in different populations. The numbers indicate that mutations distribute unequally in different populations, with the vast majority of cases detected among Ashkenazi Jews. GNOME-AD had additional nice feature by which we can calculate the variant alleles and frequency for several subsets of the data. While the default is to report statistics based on the entire data, we can restrict the analysis for non-cancer samples only. Let's take advantage of that feature to report the frequency of carriers of our mutation which develop cancer so far among the Ashkenazi Jews. The overall number of samples is 10,368 divided by 2, because there are 2 alleles in each genome, which is equal to 5,184. The overall number of carriers is 42. Let's shift now to the non-cancer dataset by changing the dataset on the upper right corner. The overall number of samples is now 9,860, divided by 2 is 4,930. The number of non-cancer carriers is 37. Subtracting the numbers, we get that among the 5,184 minus 4,930, which is 254 samples, 5 are carriers of the mutation, which is about 2%. This is more than twofold higher than the carrier's frequency in the general population. This calculation is just an illustration. In practice, there are additional mutations in this gene and many other considerations which should come to play when we want to calculate probabilities within the entire gene. In reality, about 2% of Ashkenazi Jews carry a cancer-associated allele in BRCA1 or BRCA2. The probability for developed breast or ovarian cancer in lifespan is more than 50% for the carriers. Cosmic and GNOME-AD are general resources from which we can extract also some information regarding the relation between cancer and genetics and predisposition to develop cancer. There are, however, some more oriented resources which are relevant for cancer predisposition. One of those tools is maintained in the University of Utah and available in this website. It includes internal curated databases for several DNA repair genes which play a role in inherited susceptibility for cancer, as well as collection of links to additional external resources. A good overview of the topic can also be found in the National Cancer Institute in this website. This page also includes an organized list of all hereditary cancers. A resource intended for health professionals is the Familial Cancer Database, maintained in the University of Groningen. In this resource, one can search hereditary associated cancers by syndromes. We can also search the database according to gene name. So let's search for BRCA1 associated hereditary cancers. We can see the results retrieved: hereditary breast cancer associated with BRCA1, an additional type which involves variants in non-BRCA1-2 genes. By clicking on the gene name, we get to an additional page which includes plenty of data which connect the gene and associated cancer syndromes. The most important and well-studied protein associated with hereditary cancers is the p53 protein. As mentioned by Dr. Elad, this protein is a tremendously important regulator of several pathways related to cancer. Inherited a defective copy of this gene significantly increases the risk to develop different kinds of cancers, often at early ages. Due to its importance, there are several resources which present data related to this protein alone. The p53 accounts for the Li-Fraumeni syndrome mentioned by Dr. Elad. One useful resource is the IARC TP53 database maintained by the International Agency of Research on Cancer in France. The site includes useful information regarding p53, including information on somatic mutations, germline mutations, as well as assessment of variant significance. It also includes other information such as experimental protocols and animal models. Let's examine the use of this site. We can download, for example, a complete list of information of germline mutation, disease state, cancer types, and more. This is easily done from the Download tab on the left by selecting germline mutations and TP53 germline mutations and family history. We can then open this file in Excel or other application and analyze the information. We can also analyze data within the site. For example, let's press on the TP53 germline mutations tab on the left. We can then explore the distribution of these events by pressing here on the second item here, mutation distribution. Various panels appear which include distribution of events within the gene. Distribution by codons and by the type of the change, for example. We also get a structural panel which show the variants on the protein according to the mutation frequency. We can see that the most common events occur in the DNA binding domain and include mutations of positive arginine residues, which account for interactions with the negative phosphate groups of the DNA. It is also very important and interesting to explore the tumor distributions associated with p53 germline mutations. We can access this distribution from the tumors associated with p53 germline mutations tab. In personalized medicine, we are frequently interested in knowing the particular frequency in the population of individual mutations. We can search the site for detailed information regarding defined variants. Let's go back to the main page and press on the TP53 Gene Variations tab on the left. Let's provide the protein coordinates of the variant we are interested in, such as arginine 273 cysteine, and press on Go on the right. In the results page, we see that this mutation was observed both as a somatic mutation and as a germline mutation. We have links to various resources, including GNOME-AD, which we are already familiar with. Based on GNOME-AD data, we can see that this variant is very rare in the general population. It can still act as a predisposition factor in a few families in which it does occur. The p53 individual germline mutations are not as frequent as the BRCA1 or 2 variants, at least in most of the populations. So as you can see, cancer can be also a hereditary disease, and it is very important to be familiar with the tools that help us to understand the genetics and syndromes which increase the risk to develop cancer.